Genetics of Learning Laboratory is located in School of Medicine, University of Buenos Aires, Argentina. Why do we use fruit flies?
The advantage of the flies is that we can study a large population in a very short time, and also we have many tools to manipulate their genes. Molecules and genes are very similar between flies and humans, so we can study human genetic problems using flies.
What our science is about.
Flies can learn, so we are investigating what genes are important for learning and memory. Also, we're investigating what genes are important for stem cell functions in adult tissues.
I'm studying how genetic alterations produce learning defects in human syndromes. To accomplish this, I'm manipulating genes of 3 different pathways in the fruit fly, and then I'm testing how these manipulations affect learning and memory. Moreover, we want to know if these 3 pathways activate differentially during the learning process and if they interact one with the other. Discovery of additional pathways and specific mechanisms will advance our understanding on learning in general and in human syndromes. Hopefully, these investigations will improve clinical care. Our cognitive assay consists in a classical olfactory conditioning, where flies learn to associate one odorant to an electric shock. We can perform one training session manually, or we can do repeated training sessions in an automatically way. One training session consists in presenting one odorant paired with an electric shock to a number of near 100 flies. After removing the odorant with fresh air, we present a second odorant which is not paired with an electric shock. The testing consists in putting flies in a T-maze where 2 different odorants crammed from each side and the fly have to choose.
My aim is to study how adult stem cells maintain tissue homeostasis in Drosophila's whole body and in which way this impacts on aging. For that purpose, we analyze stem cells proliferation as well as cognitive and motor aging in several fruit fly genotypes containing alterations in genes involved in stem cell functions. We are focused mainly in the protein tyrosine phosphatase SHIP2, which has been shown to be involved in both embryonic and adult stem cells proliferation and differentiation. The motor aging assay consists in evaluate the climbing capability of flies of different genotypes as they age. For that, 20 to 30 flies are introduced into vials which are placed on the device. Then, the device is tapped and the height that the flies of different genotypes climb in a time of 4 seconds is registered with a camera and analyzed on the computer.